basic concepts of what taste, color, flavor, and texture. You must have seen the product that we develop. Okay, as a scientist, you see variety of food products that you see around you. Okay, whether you take an example of biscuit, so in the biscuit itself there are variety of brands available, like you have a Parleka biscuit, Britannia biscuit, or you have in the chocolate range, you have variety, chocolate added in between, or chocolate sandwich biscuit and variety. So each one of these variety of product have a different taste. So the taste of each product is different, the color is different, and based on the color, flavor, texture, uh, a consumer accepts that product. So consumer acceptability of any product depends on these sensory things. Okay. So whenever you go to the market, you see that, suppose you have gone to a local market, and in that local market you see there are 2 3 vendors who are selling apples. Okay, obviously you will go to a place where a vendor is selling red bright color, or if your mother go to a vegetable market, if the chilies are bright in color or red in color, only based on her requirement. Some people may like red chilies, some people may like green chilies. Okay, some people may like green apple, but some people may definitely like the red apple. So there are a lot of food selection criteria which will vary from a consumer to consumer, and basically there are 5 sensory criteria one person choose on, like sight. If you take an example of sight, as I said, I suppose there are 2 glasses of juice. Okay, or if you go to a party or a conference. Okay, and in that party you will see that suppose 5 6 different types of juices are being served. Orange juice, or based on color, you do not know what are those juices. For example, one is purple color, one is orange color, but you will think that that orange color liquid is relevant to orange juice, or the purple color thing you will correlate it with the grape juice. Or if it is green color, so you will think that, oh, green color means something related to vegetable juice. So we always, based on its color, we try to correlate it with something else. Or size, suppose you have a very very big pumpkin, you think that, okay, this big pumpkin is over ripened. Or small tomato, you might think that the small tomatoes are under ripened, or they are not good quality. Apples of a very small size, you will think that they are under culled apples, which are not desirable. So a very big size, good size will be desirable. Shape is also very important. What if you get watermelon of a square size? You will think about it, you will not. Or grapes also of a very circular and oval shapes, you will consider. Or if the oranges are of different shapes, you will not like them. Or banana, if the shape is not curved, you will not like it. Shine on the apple, or shine on any product or fruit. So all these things attracts us. Texture, like from far away distance only, looking at the fresh apple, you will think that, oh, this apple must be crunchy. Or looking at the banana, if the bananas are over ripened with the black dots, you will think that, oh, these bananas are very very soft, mushy, and they are over ripened. So you will decide at the looking only, from the sight only, you will decide that the texture. Okay, when you look at the chips, you will think that, oh, they are crispy. Okay, or biscuit, you will think that, oh, they must be crispy. So based on sight, you decide its criteria. Or odor, obviously, volatile flavor substances enter into the nose, and they give the odor. And then odor attracts us. For example, if you have gone outside and there is a thela, there is a vada pav wala or samosa person who is frying samosas or vadas. So with the smell of that pakoras or vadas only, you will get attracted, because during frying a lot of volatile flavor substances are released, we have already seen, and that attracts one. And we have also the records of these substances in our mind. We correlate that. For example, uh, a tea is made with added ginger, so we know that the ginger compounds, we know the smell of ginger. So what you will do, okay, you will get attracted very easily. Oh, the tea is going on, or somebody is making a very good tea. So these volatile flavor substances, dictionary is there in our mind, okay, and based on that, we see that, oh, the chai must be of very good quality. Or suppose you are traveling on a train, okay, on specific station, you will see that the chai wala, they have added this elaichi. So there are some spaces, places of stations wherein elaichi chai is very famous, cardamom. So that odor itself will signifies, okay. So likewise, and then taste means something that you consume, and your tongue decides on it, like whether it is salt. So you must have studied that there are taste. There are different types of taste, like sweet, basic taste, sweet, salty, sour. In, I think, in third year only sensory analysis, or fourth year only there is a training of sensory analysis. And theory also. So there you will study that in detail. So today I am just giving you an overview. So basically there are four basic tastes, like sweet, salt, sour, bitter, and there is one more, it's called as origami, that is umami. Okay, so basically this taste is sometimes sensation of warm days, sensation of cold days, like mint. If you consume polo, if you consume, okay, you will get the sensation of cold. Sometimes there is astringency. For example, amla, if you eat that raw amla, there is a lot of astringency. Or if you consume, extract of black tea without sugar, you will get that astringent taste in the mouth, or there is a puckering action in the mouth. So these are all terminologies related to taste. Then sound, like just if you have those crackers, like now Diwali, a lot of snacks are made, like Shankarpara or Chakli. So somebody, if it is eating and if it is making noise, from the far away distance only, if you hear that sound, you will say that, oh, that's very crispy, or a crackling sound, or a bubbling sound, or a fizz in the bottle, which makes the fizz sound, or popping, like popcorn, they are popped, okay. So all these sensory impressions of the food, they are provided by the five sense organs, and like sight, odor, like eyes, ears, nose, mouth, skin. So basically any food that is selected is based on all these criterias. And as I said, sight, the eyes receives the first impression of the food, like for example shape of an apple or color of banana. Suppose a person is selling yellow banana or green, obviously you will get attracted to the one that is selling a good quality fruit. So color attracts, consistency, serving size, and the presence of any outward defect. Suppose there are a lot of curled fruits or curled things, or lot of insects in one grain. So you will look at the grains and you will see that, oh, this is not a good quality grain, or these are not good quality wheat. Suppose you go along with your mother to buy the wheat grains, and the wheat grain has lot of insect or lot of dust or lot of other grains, you will not buy it, okay. So because eyes receives the first impression of the food. So, and suppose juice, as I gave a very good example of the juice. Odor, again, odor is again a very very uh, different stream, a detailed stream, where a lot of research has been going on sensory analysis itself is a different area of study. People are doing masters in sensory analysis, and things like that. So odor, so basically there are a lot of volatile molecules that are present in the food system, and these molecules are capable of evaporating into gases, and these gases enter into the nose through these nasal cavities, and then uh, there is a through the olfactory epithelium, this aroma is then translated or send, the signals are sent to the brain. So basically, in the brain there has to be dictionary. Okay, so dictionary, what do you mean by? Okay, when I give training to you, maybe in third year, so as a part of practical, I have seen that suppose how do we train you for the various words, okay, of the sensory. Suppose there are 10 different aromas or flavors, like suppose mint is there, or spices like ajwain, curcumin, or jeera, or adrak. So any flavor you take, okay, so suppose there are 40 different flavors. So if I ask you to identify 40 without giving you training or without telling you that these are some of the flavoring compound, okay, so what you will do, you won't be able to identify the name of those flavor or aroma compound. You will try to correlate it to something that you have eaten or you have used in the day-to-day lives. For example, mint odor is generally correlated by the panelist or the members with respect to Pepsodent or Colgate. So they think that the flavor is coming because of the Pepsodent or toothpaste. So that's how they correlate. Or if the extract of the black pepper is there, so they don't say oleoresin. They don't say directly oleoresin. दे से ये ब्लैक पेपर है, या अगर जीरा का एक्सट्रैक्ट है तो दे विल से ओ दिस इज cumin सीड. Okay, so basically a vocabulary or a library. So our brain, day to day exposure. So for example, I'm drinking this green tea. So this green tea's flavor and odor components have been recorded in my brain. So in our day to day life, what we do through this olfactory epithelium, we get those signals of aroma to the brain, and we train our brain for these aroma compounds. Okay, so when an unknown compound is gone into the nose, you will try to correlate that compound with the dictionary, and then you will find. So volatile molecules, they are capable of evaporating like a gas into the air, and this olfactory epithelium relates to the sense. So this, if you look at this figure, when the food is entering, see, this is not just with the nasal thing, but it is also with the mouth feel and taste. Okay, so odor is associated with the nasal aroma as well as mouth feel and taste, because these aroma compound are also entering into the mouth, and from mouth there is a direct entry to the retronasal, like there is a cavity here from here. So upper palate, from the upper palate the entry is more way. Okay, so even if you consume, so these olfactory epithelium is able to identify the aroma that are released and identify. Okay, so that's how the aromas are detected. So taste, smell, they are the chemical senses. They give us information about the chemical composition of the surrounding, and we know that taste is an immediate sense, a final checkpoint for the acceptability. Suppose you go to the market, okay, and suppose there are 5 different brands of the tea. Red Label is there, Society is there, Taj is there, so many. Okay, until and unless you bring that chai at home, make the tea, drink the tea, enjoy the tea, then only you will decide, oh, this tea is better than this and this is better than this. So taste is very, very important, and it's a final checkpoint for the acceptability of the food. Smell is more distant states, allowing us to detect concentration of airborne substances. Taste and smell only separated when animals go to land. Fish and other sea creatures have one general chemical cells. Taste is not just in the mouth, okay, it is also in the nose also. Catfish have these chemoreceptors all along their body, like a catfish is like a giant tongue, okay, because and the flies that have receptor on their feet, so they can tell immediately upon landing onto the object that whether it is a thing to go eat or not. So why do we need taste? Because taste is a gatekeeper of the sensory mechanism, which is designed to taste the food and other substances before they enter into the body. And things that are potentially useful for the body tend to taste good, and things that are potentially harmful taste bad. For example, bitter gourd contain a lot of saponine, and saponines are anti-nutrition factor. Just now we saw that. Okay, so naturally we don't like it. Or tannins, okay, tea, bitter tea, black tea is so bitter in taste. We don't like it. So because the tannins are hazardous, so those things that are potentially harmful, they taste bad. So I'm quickly giving just an overview on taste, not too much of detail. So basically, if you look at the tongue, there are, I said, a basic taste. So the tongue contains many ridges. If you can look in the mirror and you can observe your own tongue, they contain ridges and valleys. They are called as a papillae. Okay. There are four types of papillae that are shown in this figure, like this. Filiform papillae. Basically, they are, see, this is the filiform papillae, which is cone shaped. So this is the filiform papillae, which is cone shaped and found all over the tongue. Okay, so these are all over the tongue, and that is why tongue looks rough. And you will be surprised, no, if you eat something hot, immediately they get burned or they get swollen, and you loses the taste, okay, ability to taste anything. And compared to all the organs in the body, tongue is the one which recovers. If you cut your tongue, you know, it's the fastest recovering organ of our human body. So fungiform papillae, they look like a mushroom shaped, and they are found at the tip and side of the tongue. Foliate papillae, there are a series of fold along the side of the tongue. See, these are the fold, like you see. These are the foliate papillae, both of the side, okay. And then these are the fungiform. Then there are circumvallate papillae. They are like flat mounds which are surrounded by trains, and they found at the back of the tongue. So all these papillae except these filiform which contains taste bud. So the very center of your tongue which only has filiform, this center which only has this filiform papillae is a taste blind. So each taste bud, like if you take this taste bud, okay, this is the taste bud. In the each papillae, you see there are taste bud. Do you see like this? So these are the taste bud. So each taste bud contains number of taste cells. So if you look at this is the taste bud, and you will see these are the taste cells which have tips that protrude into the taste pore. Okay, these are the nerve fiber, and they are connected to the nerve fiber. So suppose if you consume anything, these taste buds will sensitize, and through the nerve cells, taste cells which have these tips and that taste that are protruded into the taste pore, and their taste pores are connected to the nerve cells, and they will identify it. So basically, as I mentioned, there are four types of taste, like bitter, sweet, sour, salt. So there is a transduction occurs when different taste substances cause a change in the flow of ions. So basically, it is a flow of ions across the membrane of the taste cell. So this is your taste cell. Okay. So, and you have, suppose you come in contact with the bitter taste, you come in contact with the sweet. So each one of them have receptors on tip of the taste buds. Okay, receptor, different size, sour, H plus ion, salt, Na plus. So we will see that in detail. So this transduction occurs when different taste substances cause a change in the flow of ion across the membrane of the taste bud. Different substances affect the membrane in different ways. Like obviously bitter, sweet substances are different. They have like substances bind into receptor side which release other substances into the cell. Sour substances contain H plus ions. Salty substances break up into sodium ions. Okay, likewise. So basically, when these goes like identifies these taste buds, electrical signals are generated into the taste cells, and then they are transmitted into three pathways, like in the brain. So this is like connected through the brain. So I'm not going into all details, but if you see the chorda tympani nerve, which conducts the signal from the front and side of the tongue. So this especially nerve, uh, basically, see there are a lot of nerve. It's like a very complicated thing, but only especially this chorda tympani conducts the signal from front and the side of the tongue. Then there is another nerve called as a glossopharyngeal nerve, which conducts signal from the back of the tongue. So overall, from side, from front, from back, and from the larynx, vagus nerve. See, this is the vagus nerve which will conduct from the larynx. Then this is glossopharyngeal nerve which will conduct the signal from back of the tongue. Okay, so we have seen there are options like in the taste, the back side is for bitter, then salty, sour. If you see, there are different zones wherein you will see the different taste. Okay. Yeah, so if you look at this picture of the tongue, in the tip of the tongue it is sweet taste. From the side it is salt. Sour is just behind the salt. And bitter is like that is why whenever you consume anything, initially you will don't find any bitterness, but bitter aftertaste. Like after gulping anything, you will see that there is a bitter taste. Okay, so these are the various nerve cells that will determine salty, sour, sweet or bitter taste, and these three nerve makes connection into. So these see this nerve, this chorda tympani, all these are connected through one single nerve. Okay, and that goes here. These three nerves make connection in the brain stem in the nucleus of the solitary tract, that is NST, and before going on to the thalamus. These are the thalamus wherein it can be red. Then two regions of the frontal lobe, the insula and the frontal operculum cortex. So sweet in the front, sour on the side, salty on extreme side, bitter at the end. Okay, so these are the places. So what taste do we taste? The four basic tastes are sour, sweet, salty, and bitter. All of our taste sensation can be described as a combination of these four basic tastes. So different taste receptors, as just now we saw, and therefore different parts of the tongue are more sensitive to different taste. So experience of taste. Your experience of taste depends on your internal state. Like things always taste better when you are hungry. Like for example, that is why in the sensory analysis, you are not hired or called for sensory analysis between 11 to 12:30 because it is a lunch hour, or in the evening 4 to 7 o'clock because that time you are extremely hungry. On your past experiences, like familiar food generally tastes better than unfamiliar food. For example, ghar ka khana always is tastier than the something else. And obviously the third most important thing is your genes. People have different sensitivity to certain taste. Okay, so when I conduct the training session in the laboratory, I see that people have different thresholds, different sensitivities, different liking, and it will vary person to person. So taste experiences is also a subject to effects of adaptation. Like for example, adaptation means like coffee. in some of the countries, coffee is consumed in a highly bitter taste. Like it tastes so bitter that you cannot even take a single drop. Okay, so you're, but then slowly, if you start drinking every day, for example, Chinese, okay, Chinese food, that umami taste or that meaty taste was not acceptable. Soya bean sauce taste was not acceptable once upon a time. But when you start eating, you develop that taste. Like we experience that. Like initially, whenever I travel to different countries or stay there, you don't like that food or you don't like that taste. But over the period, when consume on everyday basis, so you start liking it. Okay, so you try to adopt. So sometimes, for example, why does orange juice taste gross after you have just brushed your teeth? Like bitter taste, we have seen. Our sensation of taste also depends heavily on smell, our texture, touch. Ever notice how food just doesn't taste that good when you have like cold? Suppose I have today very bad cold or throat pain, so I have lost all my taste buds, like they are not functioning. So this is because of the metabolic or health condition. Okay, so first taste is sweet. So sweetness is basically associated with the compound that has hydroxyl group, that is OH group, polyhydroxy compound, and many amino acids taste sweet. Some metallic salts also taste sweet. And there are unrelated compounds such as chloroform. Chloroform, if you have happened by mistake or accidentally gone in mouth while pipetting into the laboratory, you will see that chloroform is very sweet in taste. Okay, so the compounds that are associated with the sweet taste are the hydroxyl groups, polyhydroxy compounds, many amino acids, some metallic salts. Okay, so they it's always like relative sweetness is normally compared to the sucrose. For example, the sucrose has a 1 score for the sweetness, whereas glucose is less sweeter than sucrose, that is 0.76, or fructose is even sweeter than sucrose. There are artificial sweeteners like sodium cyclamate, 30 times sweeter than sucrose, or acesulfame K, aspartame 200 times sweeter than sucrose, saccharin is 350 times sweeter, and so on and so forth. Okay. So there is a theory related to this sweetness, and it is coming from the scientist who discovered it. It is called as a Schellenberg's Saporous unit theory. So basically, according to this research, the sweet molecules contain the H bonding groups such as hydroxyl and amines, and therefore you taste those things sweet. Just wanted to give you an idea that this is. Then the next is bitterness. Many plants contain molecules which are bitter in taste, like atropine, quinine, imetine, nicotine. So role plants, like it is a natural mechanism like anti-nutrition factor, but they have also pharmacological properties, sometimes desired, undesired. So animals' ability to perceive these have undoubtedly evolved to avoid eating these plants. For example, some of the deadliest poisons are very bitter in taste. Quinine antiplasmodial agent is used to prevent and cure malaria. So this malaria medicine is so bitter. There is one also herbal medicine or plant generally in the village, that plant is given to eat for those who are affected with malaria, and it is very bitter in taste. Okay, it's because of the quinine conduction. Okay. So basically, the constituents that are responsible for this category are phenyl thiocarbamide, or it is also called as a PTC4. It is one of the most notable compound in this category. Also, creatine. When I conduct this training for this creatine, we generally use for using this bitter threshold. So basically, creatine is a constituent of muscle food up to about milligrams per gram in lean meats added into soups for the taste, bitter taste. Okay, quinine is again an alkaloid that is generally accepted as the standard for the bitter taste sensation. The detection threshold means an ability of a person to detect the bitterness at the minimum level. For example, in the sensory analysis you must have studied in one of the class. So what you do, basically, you have a different solutions containing different, suppose quinine. So each solution would have a different differences in bitterness. So the level added is in ppb, okay, or ppm, okay, very less concentration. So suppose 0.01 milligram or 0.2 milligram, 0.03 milligram. So suppose there is a control. So control, I will say, okay, this is not tasting bitter. But there is a 0.01 milligram. I will still say, okay, this is not bitter. But at 0.02, I will say, oh, this solution is bitter. That means my threshold level is at 0.02 level of quinine present in the water. I was able to identify the bitterness. But for example, maybe Devraj or somebody else from your class may identify the bitterness just at 0.01 level. So the threshold is very high. That means that person is very sensitive to bitterness because that person is never consumed bitter taste. So for example, if I consume a lot of bitter taste, so my threshold will go high. I will not feel even, because if I consume a lot of bitter gourds, on like throughout the life, if one consume a lot of bitter gourd, that person will not find them bitter at all. So it's the matter of adopting with the thresholds, okay, and working with the thresholds. Okay, so detection threshold for quinine hydrochloride is about 10 ppm. So it's a normal, like for like in the laboratory when we do the sensory analysis, we for a common population, for example, from the class of 20, how many can identify the bitter taste at 10 ppm concentration? Means it's a general, for general population, that quinine hydrochloride and the bitter taste can be identified at 10 ppm. Okay. So in general, bitter substances have lower threshold than the other. For example, sugar. Suppose every day I take tea with a lot of sugar content. Okay, so my threshold for sugar becomes so high that a small concentration of sugar doesn't taste good to me. So lower taste threshold means the one that do not consume or do not get exposure. So they also tend to be less soluble in water than taste active material. Quinine is permitted as an additive in beverages such as soft drinks. Sometimes the soft drink tastes bitter is because of the quinine. So this bitterness blends well with the other tastes, provides refreshing gustatory stimulation. Purposely, some of these quinine are added into the food. The practice of mixing quinine into soft drink beverages apparently stems from effort to suppress or mask the bitterness of quinine when it was prescribed as a drug for malaria. This coffee, coca, tea, they are all contributes to the bitterness. Caffeine is added in concentration of up to 200 ppm to soft cola beverages, and much of the caffeine employed for this purpose is obtained from extraction of green coffee beans, which is carried out in the precipitation of decaffeinated coffee. Large amounts of hops, like when you study brewing, beer, some of the beer are very bitter in taste is because of the hops. Hops is nothing but flowers that are used for some special flavoring compounds. Okay, they contain the compound called as a hibulone and lipulone, and these are bitter in taste and also they have a unique flavor. So these large amount of hops are employed in the brewing industry to provide a very unique flavor. Hibulone is most abundant substance that is present in this flower of hops. And that is converted during boiling to isohibulate by an isomerization reaction. Also, grapefruit is bitter in taste, or limonene present in the orange or grapefruit causes this bitterness. So there are, scientists are developing methods for debittering orange juice, and there are immobilized enzyme being developed wherein they can bind with the bitter compound and make it unavailable. Citrus fruit also contains several flavonol, glycosides, naringine, and other predominant flavonol compound which are found in grapefruit, bitter orange, and that is why these juices, if you remove them and keep it outside for a certain time, they get come out of the cells, and therefore the juice taste, bitter in taste. Then the next taste is your salty or sour taste. So salty taste is represented by sodium chloride and is also given by lithium chloride. National policies encouraging reduction in sodium consumption have stimulated interest in food in which sodium salts have been replaced, like low sodium. There is a lot of big movement going on by FSSI wherein they are promoting low sodium or no sodium, because sodium causes blood pressure, and therefore a lot of other problems related to blood pressure. Sodium consumption have been stimulated interest in foods in which low sodium or sodium replacement by alternative substances is encouraged, particularly with the potassium and ammonium. And you know that the sodium is heart of any food. Without sodium you will not like to eat the bhaji or any food item you will not like. So salts have complex taste, mixtures of sweet, bitter. Sometimes, even if you make lemonade, salt is added, because sometimes sweet taste is enhanced by the salt taste. Okay, so this is a complex taste mixture of sweet, bitter, sour, and salty perceptual components. Or if you make lemon water, we add little salt. So sodium and lithium produces only these taste of salty. Potassium and other alkaline earth cations also produces salty and bitter taste. Chloride ion is least inhibitory to the salty taste, and the citrate anion is more inhibitory than orthophosphate anions. Okay, so these anions not only inhibit taste of cations but also contribute to the taste of their own. You must have seen there is a low sodium salt is a big launched by Marico, and there are other company who have replaced these with potassium and other salts anion. Okay, so anion taste effects impact the flavor of food such as processed cheese, where citrate and phosphate anion contained in emulsifying salt suppresses the saltiness contributed by the sodium ion. Similarly, soapy taste caused by the sodium salt of long chain fatty acid. Detergent also, they are salty in taste. Or long chain sulfates, they are all results from specific taste elicited by the anion. And these tastes can be completely mask the taste of cations. In addition to that, there are flavor enhancers that we very oftenly use. Like most commonly one is monosodium glutamate. So they simply enhance the flavors. These substances contribute a delicious or umami taste, particularly this Manchurian or Chinese food that you taste. Okay, Hakka noodle. So when used at level in excess of their independent detection threshold. So they simply enhance the flavor at level below their independent detection threshold. They contribute to desirable flavors of vegetable, dairy products, meat, poultry, fish, and other food. And which are the group of substances? They are the 5-ribonucleotides or monosodium glutamate or D-glutamate and 2 or 3 ribonucleotides. Then there is one more taste that is very oftenly seen, that is called as astringency. So basically, astringency is a taste which is related to a phenomena which is perceived as a dry feeling in the mouth. For example, if you take any red wine, or if you take any cola beverage, okay, so that exhibit astringency as well as it is bitter in taste. Or black tea, you take a black tea, okay, very good example. So there is a puckering mouth feeling in the mouth or oral tissue. So astringency usually involve association of tannin or polyphenol with the proteins in the saliva to form precipitates or aggregate. Amla, okay, कच्चा आमला हम लोग खाते हैं ना, वो आमला. So it has a lot of astringent taste. Astringency is often confused with the bitterness because many individuals do not clearly understand its nature. So polyphenols, like amla has a lot of polyphenols or tannins, cause both astringent as well as bitter. So amla tastes both bitter as well as astringent. Or tea. So astringency may be desirable flavor properties such as in tea. Practice of adding milk or cream to tea removes astringency through binding of these milk protein with the polyphenol, and they therefore becomes unavailable. Red wine is a good example of a beverage that exhibit both astringency as well as bitterness caused by the polyphenols coming from the grapes. Astringency derived from polyphenol in unripe bananas can also lead to an undesirable taste in product to which bananas have been added. Raw banana, okay. Pungency, another very well known compound that are found in several spices, vegetables, which gives a characteristics hot, sharp, stinging sensation is called as a pungency. For example, chili khao, red chili or green chili. So some pungent principle found in chili peppers, black pepper, ginger, they are not volatile, but they exert their effects on oral tissue because of the compound that are present in them. So spices, many spices like you take dalchini, lawang, yeah, mirchi, कोई भी spice ले लो, black peppers, so ginger. So these contain pungent principle that are volatile and they produce both pungency as well as aroma. And these include mustard, horseradish, vegetable radish, onion, garlic, watercress, aromatic spices, cloves. Clove contain eugenol as an active compound which is responsible for pungency. All these spices and vegetables are used in food to get the characteristic flavor or to generally enhance the palatability. Of course, usage at low concentration in processed food provides a liveliness to flavor through subtle contributions that fill out the perceived flavors. Chili peppers like capsicum contain a group of substances known as capsaicinoids, which are vanillyl amides of monocarboxylic acid having a chain length of carbon 8 to carbon 11. Whereas black pepper or white pepper, they are made from berries of Piper nigrum and they differ only in the color, okay, but they're and is prepared from immature green berries, and white pepper is made from more mature berries, usually harvested at the time they are changing from green to yellow in color, but before they become red. So the compound that is present in this black and white pepper is piperine. Okay, it is an amide. So piperine is extracted and lot of compounds they are added with this piperine. Food industry using all the oleoresins or extraction from these spices because of this pungent compound. Ginger is a spice derived from rhizome of tuberous perennial gingiber officinale roscoe, and it possesses a pungent principal compound, and this pungency of fresh ginger is caused by the group of phenyl alkyl ketones called gingerols. Okay, it's a very good compound, and nowadays due to corona and cold, this is responsible for preventing a lot of diseases. So gingerol is most active compound present in the ginger, and gingerol may vary in the length, like ranging between carbon 5 to carbon 9. There is one more sensation or taste is given, is called as a cooling. So cooling sensation occur when certain chemical compound come into contact with the nasal or oral tissue and that stimulate a specific saporous receptor, and these effects are most commonly associated with mint-like flavors, including peppermint, polo, spearmint, wintergreen, etc. So several compounds cause this type of sensation, but menthol in its natural form, that is one isomer form, is in the most active form and is most commonly used in the flavor. So camphor is also produces a distinctive camphorous odor in addition. For example, if you burn the camphor and you take the steam of the or the that smoke of the camphor, you will get that cooling sensation in the mouth. So camphor is produces a distinctive camphorous odor in addition to a cooling sensation. The cooling effect produced by the mint-related compound is mechanistically different from the slight cooling sensation produced when polyol sweeteners such as xylitol also provides cooling sensation are tested in the crystalline material. So related to smell, why do we smell? Because smell is our distant chemical sense. We can, because smell also is associated with the taste. Sometimes we can discern information about the chemical composition of substances before we taste them. We can identify. For example, pakoda ban raha hai, मुन्ना कैंटीन के वहां से गुजर रहे हैं लेकिन खा नहीं रहे, बट वी नो दैट देयर इज अ पकोड़ा और मिर्ची भजे बन रहे हैं या समोसा बन रहा है, or somebody is frying fish. So only the smell can tell you what is happening. So for many animals, smell is most important sense, although for humans, smell is not as important as say vision or hearing. But we do use smell for more than we think to guide our behaviors. Like smell is an important part of taste. Many qualities of food that we think we taste are actually a function of smell. For example, when you go to the market, you want to buy mangoes, Alfonso mangoes. What you do, you take the mangoes and put near your nose and you smell them, and you see, oh, the taste is then correlated with the smell. So in general, an overview on anatomy of the cells, smell. So you see, here is your nose, and in the nose you have this olfactory mucosa, and there is a olfactory bulb. Okay, so a closure version is this of this is like expanded, and you see these are the olfactory mucosa. This square. Okay, so these are olfactory mucosa. In the olfactory mucosa, you see these are bulbs, like you see these olfactory mucosa, there are bulbs. See, this is bulb, this is bulb, this is, so there are bulbs, and there are, to these each bulb there is one line, you see. So they are connected through this nerve, okay, to the olfactory tract, and these are called as a nerve fiber. See, these are like connected, like these are a lot of nerve fiber. You see, 1, 2, 3, 4, lot of nerve fibers, and there are receptors on these nerve fibers. See, this is olfactory. So this olfactory mucosa is a dime-sized region located inside the nasal cavity. So this is your nasal cavity. You see, from here you take the air, and here there is a nasal cavity, and is a site of olfactory transduction. Is the place where the smell is identified. So this olfactory mucosa, this one area contains receptor neurons. Okay, olfactory receptor neurons, these are olfactory olfactory receptor neurons have cilia. They are called as a little hair like projections which contain olfactory receptor proteins. Okay, so see this I told you already, these nerves. So these are the nerve fibers, you see, and olfactory receptor neurons. They have this olfactory receptor, and they have this cilia through which, and this is the mucus through which through this cilia. So here basically in the olfactory mucosa, the transduction takes place. So you have this mucus in which the odorant molecules goes, and then they are detected. Olfactory receptor proteins are attached to these neurons, and then they are then transmuted to the nerve fibers, and therefore they are detected. So olfactory transduction occurs when odorant molecule reach the olfactory mucosa, and it binds to the olfactory receptor proteins on the cilia of the olfactory receptor neuron. And when odorants bind, okay, so the odorants goes and bind to the receptor site, the receptor protein changes the shape, in turn triggers the flow of ions. Like whether, like for each taste, how there are different ions, likewise for each odor compound there are different ions, flow of ions across. Like you, if you say perfumery or flavor technology, you will study in detail, so there are, like for each flavor or aroma compound there are different ions that are responsible. So there is a flow of ions across the receptor cell membrane depending on type of aroma or odor compound, and then an electrical response is triggered in the cilium, and this electrical response in the cilium then is spread to the rest of the receptor cell, and then from there it is passed on to the olfactory bulb of the brain. And we know in the olfactory bulb there are 1000 different type of receptor protein which are sensitive to different odorants, and obviously based on your dictionary. So basically we have a total of about 10 million receptor neurons. Each receptor neuron has about 1000 similar receptor proteins, and because there are 1000 different receptor proteins, there are also 1000 different receptor neurons. So it's so complicated, and it's a very big science, and people are really studying this. So this, because odorants are such complicated stimuli, we don't yet have a complete picture of how smell is encoded by the brain. A lot of research is going on. Different areas of mucus are sensitive to different type of odorant. Smell appear to be organized, especially in the olfactory bulb. An odotope is a group of odorant that share some chemical feature and cause similar pattern of neutral firing. Okay, so as such, humans can tell the difference between 10,000 different odors. Can you imagine 10,000 different odors? But often it is difficult to name the odors, as I said before, and I have experienced also when I try to train, just with 30 to 40 odorants or flavoring compound in the laboratory, people are not able to identify. They get so confused, and they get so confused that they identify it wrongly, completely wrongly. And you can imagine 10,000 different odors what can human can differentiate between. So the training is given, like there is a different area wherein people are trained to recognize the odor, like they are called as a wine expert or perfume experts, and they are not necessarily any more sensitive with their nose, but they're just better retrieving names of smell because they have trained their memory and they have added a lot of vocabulary or dictionary. Do you know dogs can be 10,000 times more sensitive to odors than humans? And yet a individual human olfactory receptor is no less sensitive than a dog. What makes dogs so sensitive is the receptors aren't any better. They have many more of them. Like we don't have so many because we are also very good than dogs. Only difference is they have 1 billion receptor in dogs compared to only 10 billion in humans. So you can imagine, and that is why dogs are better than humans. Okay. Pleasant smells like fresh baked bread or coffee or tea are used in supermarket to encourage customer. Like in Matuga, if you go, there is a coffee shop, Annaka coffee shop. So it's so famous. If you walk from the Matuga circle, you will feel that fresh aroma of the coffee, and people will go just there and buy it because of the aroma. Color is another most important factor that affects on our life. In fact, it is the most strongly influencing thing. Like one cannot wear every day same clothes, same color clothes. So furnishing in our homes, or color that we give to our home, or whatever you choose, it's a liking. Like some people choose everything blue or everything black in their home, or if there are kids, everything becomes pink in the home. So it's like color influences a lot. Okay, also the gardens that we decorate with different foods that we consume. So most of our color choices are unconscious, but much research has identified that the color of our surrounding affects our mood, affects our perception. That is why when you live in a greenery, you don't feel stressed. Okay, you don't feel depressed, you don't feel lonely. That is why in the older days the saints and more saints, they went to the forest to do meditation or to do to obtain the gyan and other things. So color of our surrounding affects our mood and quality of life. So everyone is sensitive to the color of the food, and obviously in the food, the appetite is stimulated or dampened in almost direct relationship to the observer's reaction. For example, initially I gave red apple. The color of the red apple indicates that they are fresh in color, or red chili indicates that they are too spicy. But there are variety of chilies that are available which are bright red in color but not at all chili, like spicy, like Kashmiri chili or Degi chili. So there are natural colors. Significant development have occurred with the natural colors since their wide commercialization, and nowadays people want natural color instead of synthetic colors in the food items. Okay, so a lot of studies going on pigments like curcumin responsible for bright lemon yellow color, and it can be extracted or obtained from turmeric rhizome, and it can be added into bread and the bread will have a yellow color. Or lutein, which is a golden yellow color extracted from marigold or alfalfa, and it is an oil soluble carotenoid pigment naturally present in many foods. uh, Natural mixed carotenes like palm oil has these carotenoids. They are golden yellow to orange color, or big seed or non-big seed. So there are bush seeds, and they gives an orange color. Or capsanthin, that is paprika, has a reddy orange color. Lycopene from tomato, which has orangey red color, or orange to red color, is extracted from the cochinelle insect, which is obtained in Peru, and it is because of the carbonic acid, which is red color. Or betanin is present into the beetroot, or anthocyanin present into red cabbage, or pomegranate. Or chlorophyll present into vegetables, green leafy vegetables. Or crocin present into saffron, or gardenia fruit is in the yellow color, and so on and so forth. And we know that the color influences the flavor perception. The consumer expect red drink to be strawberry or raspberry or cherry flavored, or yellow to be lemon or green to be lime flavored. So the impact of the color on sweetness perception has also been demonstrated. Like beta carotene or riboflavin are not only colorants but they are also nutrients. We have seen already the role of beta carotene, like it's a vitamin A. So the absorbance of this color is because of the wide electromagnetic spectrum, and each of the color has different spectrum. Like if you see, you have these radio, microwave, far IR, IR, near, visible, and UV. So we always work with these visible and UV in the visible range, which varies from 380 to 780 nanometer, and you see this violet, blue, green, yellow, orange, red. Okay. So the colorants have been classified like in food and additives. You will do them in detail, but just there are certified and there are like natural. They don't need certification, like anthocyanin, juice concentrate, annatto extract, and natural identicals such as carotene. Okay. So there are most commonly pigments seen in animal and plant tissue. One is hemi compound, and pigments are not there, but for the study, because you will be doing these pigments in some other chapter. Chlorophyll pigment is responsible for green color of plants, and because chlorophyll absorbs, I remember somewhere I had mentioned that it has this porphyrin ring. I think folic acid. So its unique ability to convert energy of the sun through photosynthesis is used to make carbohydrate from water and carbon dioxide. So there is a common relationship between this chlorophyll and hemi. So the both of them have this pigment. So in human blood, that hemi pigment is there, which has this iron pigment. So these are like hemoglobin molecule which has this iron, you see. So the myoglobin forms the pigment which is responsible for making the meat red. So the color that meat takes is partly determined by so basically this meat, if some of you consume the red meat, you will see that this meat changes color during processing. So cooked meat is brown because the iron atom that is present at the center is oxidized state. So this gets oxidized, and therefore there is a change in color because it has lost an electron and is now coordinated by a water molecule. So generally meat has lots a lot of water. So under some condition meat can also remain pink all throughout the cooking, because if you add certain salts such as nitrites and nitrates, so it will remain pink because the iron atom is bound to now nitrogen oxide, and then it will not get oxidized or removed. So nitric oxide, like a true of example like corned beef or cured hams, they remain pink in color. Another example is your grilled meats. They can also remain pink because of the on a pink or smoke ring that comes from the iron binding to a molecule of a carbon monoxide, like a smoke. In the smoke, there is a lot of carbon monoxide. Okay, so this raw meat is packed in a carbon monoxide atmosphere, also shows this same pink smoke ring due to the same coordination chemistry. And this chlorophyll pigment is responsible for the green color. So this magnesium, which is there at the center of the molecule, contains special ring-shaped formation which capture the this ring-shaped thing which captures the wavelength of light, but green is not captured, and the overall effect of the green color is reflected back to our eyes. And the magnesium that provides a highly alkalizing effect on the body. A lot of efforts are being done by the scientists to preserve the green color of the vegetable by cooking it into different solution, like when green vegetable are cooked, the magnesium removed from the center of the molecule and it is replaced by the hydrogen atom, causing some vegetable turned into olive green color. So there are salts that can be added. They are called as acid neutralization to retain the chlorophyll. So in these, alkalizing agents are added, color is retained, like calcium oxide is added, sodium dihydrogen phosphate is added, and the pH is maintained so that calcium or magnesium hydroxide can take place, and here therefore the magnesium atom remains as such. Carotenoids, we have already studied a lot about carotenoid. Anthocyanins are the compound that are present in the pomegranate. They are responsible for wide range of colors in plants, including blue, purple, violet, magenta, red, orange. The word anthocyanin is derived from the Greek word anthos, means flower, and kainos means blue, the blue flower. So these compounds have attracted the attention of chemists because of the lot of health promoting effects these anthocyanin are given. Okay, so these a lot of techniques are identified to extract these pigments and used as a colorant in the food system. So texture is another very important character of every food that we eat or drink. As I gave you an example, a chocolate giving bubbling a noise in the mouth, or a crackling noise in the mouth, or chips, if they are soggy, you will not like. You want the chips to be crispy. So many people, the texture is even more important than the taste. Think of all the ways we describe a food's texture. Like for example, the student in the class could probably come up with a 20 or more words just to describe the food in their lunch boxes, like crunchy, like if you carry the carrot, chewy, chapati, chewy, crispy, suppose you bring krakra, juicy. Okay, sometimes apples are juicy, squashy, runny, solid, hard, soft, soggy, firm, creamy, fatty. So these are all related to texture and so on. So the components and ingredient within food process determine the texture. As I said, the melting property of some of the cookies is big. Mouth melting property of some of the cake is because of the special hydrogenated fat. Okay, or chocolate has mouth melting property because of the fat, or even ice cream melts in the mouth is because of the some of the ingredients. So take the example of making a fruit smoothie, which is easy to make at home in a blender and found on many supermarket shelves. Depending on type of fruit, how pulpy, how puree the fruit is, how much water is there. It will determine how thick the final smoothie is. For example, smoothie made from banana or smoothie made from cherry. So cherry won't be too pulpy. Okay, so banana will give you a viscous thing. Okay, but watermelon may not give you the viscous smoothie. So most commercial products proudly claim to contain only fruit. So all the texture comes from the food. So these are various terminologies, being used to attract the consumer, and they play a lot with the texture. So different smoothies can vary in their thickness. If one is so thick that it requires a spoon to eat, then it will give an idea that it is really a good quality. So generally texture is related to sensory property. Texture can also be used to determine chemical or physical property of the food products that contain more information about chemical or physical property than color and size. For example, apple. The freshness of apple can be correlated if the apple is crunchy, or banana is like a hardness, X hardness, or bread. So bread has a smoothness or softness initial days, but later on it becomes hard. So the texture can give you chemical or physical property. So textural property of food product will change during the storage. For example, bread becomes hard, or banana becomes over ripened, or meat becomes like spoils, or fish texture changes. So the importance of texture in the overall acceptability of food varies widely depending upon the type of food. For example, texture is very, very critical. Food in which texture is the dominant quality characteristic, such as meat, as I gave you example, potato chips or corn flakes or biscuits. So what is important here? Food in which texture makes a significant but not a dominant contribution to the overall quality. Like fruit, as I gave you example, vegetables like bhendi or lady's finger, if they are fresh, I am able to cut them very nicely. But then if it is over like old days zucchini or older lady's finger, I won't be able to cut because already the starch has converted into cellulose. So I can say that the chemical changes have taken into that zucchini, and entire starch has been converted into cellulose and fiber, and it is not now good. Or cheeses, some of the cheeses, camembert or other. So bread, as I said, the firmness of the bread increases during the storage, and so on. Minor like food in which texture makes a negligible contribution, like beverages or thin soups. Okay, obviously thick soups. So there are common vocabulary like crisp, dry, juicy, creamy, crunchy, chewy, smooth, hard, tender, softer, springy, coarse. Like salt is like texture of the salt is coarse, or rava is coarse, firm, slippery, greasy, tough, sticky, watery, fatty, and so on and so forth. Okay, so there are a lot of analysis could be done on texture, like there are machines that are used for measuring the rheological property or textural property. Like they can be like sensory analysis, like you can just take the bread, put it in your hand, and see whether it is spongy or soft, or you can put it in the mouth and you can see whether it is soft or hard, or okay, there are texture analyzer. You can measure using force required to tear some specific chapati, or compress the bread, or break the bread. So rheological parameters can be checked in terms of Young's modulus, shear modulus, which is like then directly correlated with the hardness. Young's modulus has mouthfeel. Poisson's ratio is related to chewiness, or viscosity is directly related to gumminess, or stickiness, or loss compliance can be related to adhesiveness. So there are a lot of textural parameters that are analyzed, and our student, whenever they are developing any food product, we do analyze all these parameters, but simultaneously we also do sensory analysis, and we try to correlate hardness with cohesiveness, or viscosity with the stickiness, or elasticity with some chewiness or adhesiveness, and so forth and so forth. Okay, so these are very, very important parameters. Like even geometric characteristic can be converted to texture, like particle size or shape can be converted to gritty or grainy or coarse, or shape like fibrous beets are fibrous or cellular or crystalline, as I gave you example of lady's finger or zucchini. Okay, and they can be correlated with the water activity, like for example, fresh apple or